Hello, hello, seconds out, seconds in, second servings. Welcome to the Famelab International, also known as International Famelab, second Tremi final. So I'm still Quentin Cooper, still tweeting us at QWERTY, still host, still crazy after all these years. Who was at Tremi Final 1 mere hours ago? So get the lights up. Decent percentage. Okay, who at least has previous with Famelab, having been to a Famelab before? And who has wandered in not really sure what this is? Oh, there's always a couple. Good, excellent, glad to see you. Okay, let me explain. Now, on second thoughts, let's not bother. You'll get the hang of it. It's like any good game. You'll get the hang of it as you go along. Basically, each Famelab gets 3 minutes to try and convey something amazing, amusing, scintillating, stimulating, mind-blowing about science in a way of their own choosing. The rest, you'll figure out as we go along. Suffice to say that it started right here, more accurately over there in the shrubbery, about 10 years ago. With the help of the British Council and eventually NASA, it has spread to become the world's biggest science communication competition. And as of Friday, there will be 1 billion more potential entrants as a very large new country joins the Famelab fold. Woo! Okay. Can we just practice that? Thank you very much. I wanted a woo, actually. Can we have a woo? Woo! Ah, that's more like it. Thank you. So what you are about to see are 9 national champions who've won through their own heats and national finals, and part of their prize was to come to Cheltenham to take part in this Famelab International Tremi final, but it also as a kind of parade of talent. The fun bit is what they're about to do. Now, I could tell you more, but as I say, I think you'll figure out as you go along. Tremi Final 1 has already sent through the winners from Italy, Poland, South Africa, Spain, and the UK. It was supposed to be 3, maximum 4, but we had what they call a wardrobe malfunction with the voting system, so somehow we've ended up with 5 people going through from Tremi Final 1. But who from? Australia, Azerbaijan, Brazil, Cyprus, the Czech Republic, Kazakhstan, Qatar, Thailand, and Vietnam is going to join them. That was in alphabetical order. There is no favoritism here. Their fate rests mostly in the hands of the 3-headed beastliness that is our judges. First, our chair, who after studying computer and cognitive sciences founded his own communication consultancy firm and now organizes and galvanizes a whole range of science popularizing events, mostly across Italy, and he's involved in Famelab Italy as well. Applause, please, for Mattia Crivellini. Mattia. Next, he was chair this time last year, but he's taking a supporting role now. He's still a biologist, broadcaster, professor of science communication at the University of Gloucestershire, and an entoetymologist. He likes studying the origins of insects. Adam Hart. Adam. And in her own words on Twitter, she's a neuroscientist, bookworm, and landlocked beach obsessive. In more formal terms, she's Royal Society Research Fellow and Associate Professor in the Department of Psychiatry at Oxford, Liz Tunbridge. So Mattia, trying to pick 3 from 9 people who are already champions is an almost impossible task. Why on earth have you agreed to do it?
Because we are looking for passion. I think that we will find passion in these guys, and we will try to get the best 3 science with passion today.
All right, so although the official Famelab criteria are content, clarity, and charisma, the particular Italian twist is forget that, it's all about passion.
Exactly.
Okay, it's all about passion. Liz, as judges, you kind of have to be a kind of hive mind, making a collective decision. We don't know who does what. But we know from Mattia he's particularly going to be looking for passion. What's particularly going to make the difference for you? Because these are all champions. You're looking for something to lift the champion above champions.
Yeah, I think someone who brings an excitement to science is always appealing from my point of view. So somebody who can communicate clearly what they want to say, but really get the audience engaged and excited about what it is that they're talking about. I think that's what I'm looking for.
And Adam, almost half of our 9 finalists have come more than 6,000 miles to be here. Is that something you factor in when making your decision?
I don't think so. I think no matter where you come from, what you do, it's the right answer. We want to be mesmerised by what we see. We want to be looking and thinking about nothing else than what's in front of us at the time when they're talking, I think.
Good. And just finally, I mentioned content, clarity, and charisma. Can you just say a little bit more about them, Mattia, for the benefit of the new people here? When you say content, what do we mean content? What do we mean clarity?
Yeah, for content, we mean the science behind the story that these guys are put on the stage. Clarity, everything, everybody can understand, need to understand science. So this is another important things, and also charisma. When you're on the stage, when you try to say something, charisma is a fantastic view to get an emotion to the people. So
brilliant. And whoever you decide on, however you decide, you have to do it decidedly fast, and you have to break hearts at breakneck speed. So can we please preemptively thank our judges for doing a really tough job? Now, of the 3 Tremi finals, this is the most far-flung. Between them, our 9 Famelabers have travelled more than 40,000 miles to be here, and they'll have to do the same to get back home as well. That is nearly twice round the equator. And remember, unlike our judges, our Famelabers are all winners, and they're almost all presenting in what is at best their second language, so that makes what you are about to see all the more remarkable. Now, the order that they go in is random, although hopefully what they say won't be. And the first name out of our hat/random number generator/quantum computer is that of our Famelab champion from Vietnam, Thatch Pham, apologies. Thatch is studying cell biology in Hanoi and says he's been seduced by the unlimited exciting things about cells. He thinks every single cell has a story to tell, be it romance, comedy, or horror. And given that we've got something like 37 trillion cells in our bodies, that makes 37 million stories. It's going to be tough managing that in just 3 minutes, but he'll give it his best shot. It's always tough going first, so we can have wild applause, please, for our Vietnam champion, or Vietnampian for short, Thatch Pham.
Like many young men of my age, I wish I have a girlfriend. But I don't. Hopefully, I've read that girls are very attracted to boys with pets. Yeah, I got a cool pet. My pet, not the puppy dog, or naughty cat, or bunny rabbit. It's very tiny and sexy, and it possesses a magical power that can help to cure HIV. Sounds amazing. And what is my pet, and how they can do that? Yeah, the scientific name of my pet is hematopoietic stem cell. People call that blood stem cell, but I love to call that blood stem pet. Yeah, it's a pet name. And come back to, in 2006, hope came with the Berlin patient, Timothy Brown, who had been cured HIV by blood stem pet transplantation. Blood stem pet from donor to the transplant to the Timothy mutated in the CCR5 gene, which encodes to the protein, the HIV receptor protein in the surface of white blood cell. And let's enter the Timothy body and discover what happened. After transplantation, Timothy body have 2 types of blood stem pet. That is Timothy blood stem pet and donor blood stem pet. And Timothy blood stem pet will go through puberty and mature to become a white blood cell, a very attractive white blood cell, because it has a very beautiful protein. Oh, that's the receptor, HIV receptor protein like this. So, tragedy always comes along with beauty. HIV will recognize us and enter the cell through the receptor and multiply in the cell. The cell will explode. White blood cells cannot do their function in the immune system. This will lead to die. But donor blood stem pets will multiply and also go through puberty, and they will mature to become a white blood cell. But this time is different, because it mutated in the HIV receptor protein, so this protein is adventure like that. So HIV are unable to recognize, so they cannot enter this white blood cell. So white blood cell will can perform their function in the immune system to cure the infection, to cure the disease. So this is how the Timothy can be cured HIV. Each year, there's millions of people are painfully waiting for that because of HIV, but not that. But more painfully, each year, it's a social stigma. HIV patients are treated unfair in the workplace, especially children. They have no fault, but they're not welcome in school. So you can donate your blood stem pad, and we can modify them and train them and modify them to become a mutated one, and that brings hope to the children and others. Maybe they can change the world. And if anyone of you here, especially girls, want to pet your pet, we can hand in hand together petting our amazing pet. Thank you.
First of all, can you see your T-shirt better?
Yeah, can we see your T-shirt? I can't see it.
Just looking for Famelab, I can't keep count.
Okay.
It says in your little biography that some years ago you were seduced by the unlimited exciting things about cells. I just wondered if there was one particular moment where that seduction took place.
Sorry, I can...
It says in your biography here, it says
Yeah.
that you were seduced by the unlimited exciting potentials of cells.
Yeah, right.
I just wondered if there was one specific moment when that seduction happened, when you were won over by the power of cells.
It happened that throughout this university, I read a lot of book about the cells, so I think, wow, it can change to this one, but it can also can change to this one. And maybe some cells always come along together and support together, and maybe we're a good couple. And maybe when I can see under the microscope, it's very interesting about the cell. That's happened, that moving. That's interesting.
So I think it's fascinating what you were talking about. How viable do you think this will be as kind of a widespread treatment going forward? Do you think this is something that could take off and be used easily, or is it quite an involved kind of treatment that you think you're looking at?
Leukemia?
Yeah.
I think people should donate the cells. So the patient must find the matching people to transplantation, and so that's very meaningful to donate the cell to not only to treat HIV, also other blood disease like leukemia.
Okay, I have to leave it there once more for the Vietnamazing, multi-hatted Fat Fam. And to paraphrase a line from an American TV show that was on before even I was born, there are 37 trillion stories in the naked body. This has been one of them. Second, although as a semi-professional athlete, he's more used to coming first, we have our Famelab Cyprus winner, Constantinos Timinis. When not training for track or field, Constantinos is a master's student in molecular and applied physiology. He tells us his research is entitled Accurate Estimation of Refrigeration Time of Pork Using Ramen Spectroscopy. I prefer pork with ramen noodles, but each to his own. Now, he's a great believer any scientific subject can be explained simply and quickly by relating it to things in our daily lives which work in exactly the same way. Sounds great in theory, but what about in practice? We're about to find out over the next 3 minutes with our Cypriot laureate, Constantinos Timinis.
If every time you hear of the word hero, Superman comes to your mind, then it's time to think again. Let me introduce to you the tiniest superheroes ever, nanobots. And no, they don't find crime in Metropolis, but instead they are injected into your bloodstream and fight for your health. Nanobots are in fact computers in the size of molecules, and every part of them, from hardware to software, is actually made of DNA. And this is very important, as this is how they can recognize other molecules and interact with them. Now, think about all those cancer drugs when they interact with a region of healthy and non-healthy cells. They kill all of them. Have you ever imagined Superman punching everyone, including innocent civilians? Well, this is almost what a cancer drug does. But nanobots can be programmed to recognize and punch only the bad guys, which in the case of cancer are tumor cells. So we might not need new drugs. We have powerful drugs already. We just don't know how to use them right. This is a problem, and nanobots are able to solve it. But how can they recognize target cells? They use a molecule lock, very similar to a computer login system, with the molecules behaving as the digits of the password. They begin their journey in an off state, and they safely hold inside any drug they carry on without causing any damage to healthy cells. But by recognizing the right molecules combination, they switch to an on state and expose the drug to the target cells. Think about all those collisions happening in therapies where multiple drugs need to act simultaneously. We can have two types of nanobots, one with drug A and the other with drug B. Timing the activity period of each drug allows them to operate without colliding. It's like having both Superman and Batman in the same town or in the same movie, but only one of them fighting at every scene. And there are even more amazing things nanobots can do. Using shake hands technique, they form physical bridges, which allow them to extend from the one part of an injured tissue to the other, as Spider-Man does using his web to transfer between buildings. Furthermore, nanobots are able to count themselves and switch to an on state only when they reach a certain number, allowing in this way any drug to be effective. Imagine Spider-Man waiting for Superman and Batman to join him before any fight. I know, they could beat anyone. And though all these superheroes are part of science fiction, nanobots are not. So if they seem as a distant future, think again. The future is always beginning now.
Great talk. Can you give me your vision about the next 10 years in this field?
In fact, Professor Rido Bachelet, the brain behind all this, he found no reason why nanobots can be applied in the next 5 years. They can already identify 12 types of cancer. And I think it's probably they have a brilliant future on it.
Okay, just for me, make a decision, DC Comics or Marvel?
This is nanobots, and for nanobots, so no comics, no Marvel.
Third choice. Okay.
So I was really interested in, you were talking about cancer. Do you think these nanobots have potential for treating other diseases as well?
Of course. I take cancer. I like cancer, but they can also treat leukemia, solid tumors with cancer, diagnose when diabetes sufferer needs his insulin, and perform surgeries in injured tissues. So it's what I told for Superman, from the one part of an injured tissue to the other. So they are going to have many applications as they are located in the region they need to act autonomously. So except from the delivery system, yes, they can also perform operation surgeries in their own. And after they done, they disintegrate and get out of our bloodstream.
Drugs often come with side effects. Are there any side effects? I'm being told to keep it quiet. Are there any side effects of nanorobots? Quickly.
Keep it quiet, keep it short. That's been short, not quiet.
Short, quiet.
Yes, actually, this is why they are already live. But sorry. They are already checked in cockroaches' bodies, and we have great results. So I think in the next 5 years, we are able to overcome any side effects. So they are biological systems. They act like nature does. So we hope that no side effects will be.
We hope so. Okay, tiny robots, but huge applause, please, for Constantinos Timinis. And for the anagram fans, Constantinos Timinis is an anagram of instincts to insomnia, which clearly does not apply. Two presenters so far, bit of a Rips t-shirt theme developing. Be interesting to see what happens when we get to some of the other contestants. Now, we've already been over 6,000 miles to Vietnam and 2,000 miles to Cyprus. Now we're going 6,000 miles in the other direction to our Brazil and our Famelab winner there, Jackson Itikawa. Jackson grew up in the Brazilian Amazon, which he describes as not the most ideal place to pursue a career in science. Nevertheless, he did, thanks, he says, to encounters with various individuals whose passion for maths and science encouraged him to keep learning. Now based in Sao Paulo, the largest city in the southern hemisphere, fact fans, Jackson is a postdoctoral fellow working on one of the great maths problems published by David Hilbert back in 1900 that is still unsolved. One day, he hopes he might crack it, but for this afternoon, he'll settle for something mathematically much simpler, one of the places in the final. Please give an equation ovation to our Famelab Brazil winner, Jackson Itikawa.
Oops, I'm sorry. We are so used to send and receive messages using our mobile that we don't notice that we do that in the most inconvenient moments. Sorry. Well, another thing that we don't notice is that technology behind mobiles and internet is based in mathematical concepts. Today, I want to talk to you about one of these concepts, prime numbers. A prime is a number greater than 1, which has no positive divisor other than 1 and the number itself. 2 is a prime because its divisors are 1 and 2. 3 is another example, but the next number, which is 4, is not a prime because its divisors are 1, 2, and 4. Now, any number is built by multiplying primes together. Numbers are the codes of mathematics, and mathematics is the code of science. Then the primes somehow are in the core of both mathematics and science. But primes are well known since their antiquity. Actually, Euclid proved that there are infinitely many primes, and it was more than 2,000 years ago. But what Euclid never thought about is that primes would be at the heart of the codes that protect our world's cyber secrets nowadays. Our WhatsApp messages, our credit card numbers, the Scotland Yard's files, all this is protected by cryptography, which is essentially based in prime numbers. When we put our credit card number in a computer in order to buy something online, our computer receives a large number, which is called a public key. Then it performs some calculations with this number and the credit card number, and the resulting encoded message can be sent across internet because the encoded message can only be decoded with the primes that divide this large number, which is the public key. The basic concept behind it is quite simple. Although it's very easy to multiply two primes, no matter how big they are, it's virtually impossible to pull them apart. Now, if someone figures out how primes are hidden among all other numbers, the whole idea of cryptography might be compromised, since you can use this pattern of distribution of primes in order to find the primes used to make these public keys. Fortunately, this is one of the big mysteries in mathematics, and a 1 million prize has been offered to anyone who can solve this. Now that you guys know about prime numbers, would you like to sort this enigma out? That's it. Thank you.
So do you think it's soluble? Do you think anyone will ever crack it?
Well, people say that with the quantum computers coming out, their enormous amount of capacity of processing data, they will be able to figure out this public keys strategy of doing cryptography. Then we would have to figure out some other way to make secure our transactions through internet.
I'm hoping that somebody's working on that now, as well as working out how to crack it, right?
Yeah, probably.
My bank account will be good, so thank you.
Okay.
We are fine.
Okay.
Thank you.
Thank you.
Very much.
Judges, you only have to give me 2 minutes of questions. These guys, they've got to memorize 3 minutes. It's terrible. That was a prime example of Brazilian brilliance from Jackson Itakari. He deserves one more round of applause for that. Thank you. Well, coming up next, we have the nearest thing we have to a local candidate in this second Tremi-final, because they've only come from the Czech Republic, which is a mere 700 miles away, and it's Eliska Selinger. Now, Eliska's a medical student specializing in immunology and the epigenetics of cancer, or as she puts it, a young researcher in love with the poetry of epigenetics and caught by the web of cancer immunology. Eliska's way is definitely better. She also says she likes her coffee strong, her wine dry, her chocolate dark, her cuisine spicy, and her Famelab presentations, well, you're about to see as she talks about 50 shades of the epigenome, Eliska Selinger.
Shock. A bee larva ate royal jelly and became a new queen bee. Horror. Did your parents suffer from famine? If so, you will die of a heart attack. All these could be headlines of tabloid articles about me. I'm DNA methylation, one of the ways by which your cells determine which genes they need and which they don't. It's because of me and other regulatory mechanisms that people were able to create a whole new branch of science. Epigenetics. Epi, or above. So it's obvious who is the dominant one. To begin with, imagine DNA, a long chain of letters or bases connected to each other. From 20 to 30,000 of human genes written in DNA, I command which one should be dormant. So it's impossible for cells to use them. And the reason for all of this is love. I love corsets. I love cuffs. I love CHAINS. I love C's. C for cytosine, one of the 4 bases or letters DNA is made up of. In the genetic code, I find the right cytosine and chemically bond to them. Surrounding DNA then fastens to us too and hugs so tightly, the cell cannot see what is written in the gene. Do you like bondage? I do. And it seems that my BDSM love story could last for 2 to 3 generations. But usually, my affair with the gene is not for life. When we break up, my X gene releases his hold, and the cell can see again what is written in his DNA sequence. My cytosine hookups and breakups are probably controlled by environmental influences, like famine or the royal jelly, which contains substances disturbing my cuddling ability. So, remember, your parents maybe didn't just give you their DNA, but also a dash of their life experiences which they epigenetically saved on it. How nice of them to leave you bits of advice on what to do with that huge amount of genetic information until you get your own epi-mind. Thank you, and maybe laters, baby. So, questions?
Well, thank you. Thank you very much for your interesting point of view.
Thank you to listen.
And when did you get this idea? In your country? When did you get this idea? I mean,
I was thinking about DNA methylation, condensing DNA, bondage DNA, so just idea.
I wonder what you thought last week. Richard Dawkins came out and said that he thought epigenetics was a flash in the pan, that it's a short-lived phenomenon. It's not really of great evolutionary importance. I think your presentation of it was rather more entertaining than his, but I wonder what you thought of that kind of thought about epigenetics.
Once more, please. I like to listen loud to music. So.
Epigenetics, some people say, is not a particularly important evolutionary phenomenon because it's not very long-lasting. I just wonder what your thoughts on that were.
I don't think so. We know that there are some metastable epi-alleles that probably could be transmitted through the generations. But of course, it's much more complicated, and we still don't understand the language of epigenetics exactly. But for example, we have epigenetics treatment for not cancer, but hematological disorder yet. It's non-specific, but it's inhibitor of DNA methyltransferases, which are the enzymes which makes DNA methylation. So I think in, for example, general medicine or medicine, this is my field, it's promising. It could be promising. We have clinical trials which try to combine epigenetic treatment with other treatment like chemotherapy or immunotherapies to improve the results and so. But we will see. It's really, really complicated.
Okay, whip up some frenetic epigenetic applause, please, for Eliska Selinger. Strange the panel didn't have a shortage of questions for Eliska, did they? Yes. If you thought S&M stood for science and mathematics, think again. Right, okay. And appropriately for somebody who likes the poetry of science, Eliska Selinger has the fantastic Famelab anagrams of rises angel-like and girl seeks alien. Right, we're halfway through this tremendous Tremi final and almost halfway through all 3 Tremi finals today. Next we have someone who's come almost exactly 6,000 miles to be here, our Thailand Famelab winner, Khao Thonsombun. Now, as a child, Khao knew exactly what he wanted to grow up to be, a singer or maybe a film director. But he says, the success of my grandma's total knee replacement 15 years ago inspired me to become a biomedical engineer to save other grandmas' lives. Now, some may have thought he was daft to decide on a career for such a daft reason. There's a bit of a knee-jerk reaction. But Khao followed through. No, never mind. Khao followed through, and having got a biomedical engineering, PhD in biomedical engineering from Cambridge, he's now back in Thailand as a researcher at their National Centre for Genetic Engineering and Biology. When not saving grandmas, Khao is out to change public perceptions of science across Thailand. He says that instead of it being seen as difficult and distant, he wants everyone to realise it's fun and relevant. And do you know what could help with changing those perceptions? That's right, the next 3 minutes. Bang your hands and slap your knees for the Thai typhoon that is Khao Thonsombun.
Thank you. "15 years ago, my grandma couldn't walk because her knee got a problem. Knee is where the upper leg and lower leg bones join together. For young people like us, we all have the white tissue here called cartilage that prevents 2 bones from hitting each other when you walk. But my grandma was old. She didn't have it. So I asked surgeon, what would she do without you, cartilage? Surgeon said she needs to get her knee changed. Her bone will get drilled in, then put steel in. This hurt like insane. I thought this is over, but I was so wrong. It lasts for just 20 years long. I was sad when I knew that the metal knee would cause my grandma's bone a problem. Bone is so smart. When you gain weight, it can sense, ooh, heavy. I need to get thicker to carry this belly. But when you lose weight, it can sense, oh, not much weight to carry. I can get leaner and chill out a bit. So when surgeon put metal into my grandma's bone, most of her weight goes to the metal because it is much stronger, and her bone will carry only small weight and become thinner, leaner. Over time, her bone around the metal will disappear, and her metal will be floating and shaking because nothing is surrounding it, and she wouldn't be able to walk again. Imagine if you had to get a metal knee when you were young, like 35, you may have to get another one and a more painful surgery when you're 55. Doesn't sound good. So scientists are trying to regrow cartilage from patients' own cells. But it's not easy to control them to become cartilage. We have to feed them special food and grow them in a specially designed jelly that has similar softness to the cartilage to guide them. Because when cells touch the wall of this jelly, it can sense, ooh, soft like cartilage. All right, I grow into cartilage. Lots of clinical trials in humans are being done, and we hope that one day we'll be able to grow cartilage so patients don't have to experience the second knee surgery. Because no matter how hard they try to avoid it, they can't. Because steel is stronger than bone, so bone gets weaker and gone and gone. Thank you.
Awesome, awesome. Can you tell me more about your decision to come back to your country?
Because my family is there, I have to go back to live with them. But it was privilege. I got a scholarship from the Thai government to study here from high school to PhD. So I want to go back and improve my country.
You said that you want to change the public perception of science in Thailand. I wondered if you had any ideas about how you might go about doing that.
We have many projects coming up, but right now I'm trying to give talks, inspirational talks to children in every single science festival, because I want to change not just the perception of children, but more importantly, the perception of their parents. Because in Thailand, most parents want their children to become physicians because it's a great job. So I want to show them that scientists like it, have a good salary, and we can live well. And I also plan to launch a YouTube channel that would tell citizens about the news, bring science to the current news, what the hot topic in Thailand right now. Just because they have many pseudoscience, and that's the best way to reach greater audience.
I'd like to congratulate you both on your musical timing and also your impeccable timing with the squeaker, which was really very impressive. I was just wondering if you could talk about how far in the future you think the kind of the cell-based developments will be.
If you ask me to make a prediction, I would say in 5 years or 10 years' time, globally we would be able to, hopefully, we'll be able to use the cell-based therapy to help patients in orthopedic first. I think bone and cartilage, lots of research are being done on bone and cartilage because we're getting into aging society, so that's why we target bone and cartilage first. And I'm pretty sure in 5 years, 10 years' time, we can do that.
So I need to start growing my cells now, is what you're saying.
You can, but you better take a good care of your knee because you're still young. But we all have time to take care of them.
Take-home message. He's given a great changing perceptions, so give a great reception to Tao Kontonboom. I've had 3 knee operations already, so I've only met him sooner. Now, up over from down under, we come to our Aussie champ, Erinn Fagan-Jeffries. As a child, Erinn used to sort clothes pegs by colour on the washing line. This was one key reason she grew up to be a taxonomist. It's not the only reason, but the others are classified. Thank you very much. It's my favourite joke. Her particular interest is in entomology, which Adam should be interested in, so much so that Erinn runs a citizen science project called the Caterpillar Conundrum, where she gets children to inspect insects, then pupate and turn into butterflies. I may not have got those details entirely correct. Formerly a member of a science circus that toured schools and communities across Australia, she's now returned to the lab at Adelaide University to study for a PhD. This afternoon, though, she's back on the high wire for the next 3 minutes. So please welcome Erinn Fagan-Jeffries.
I'm about to ask you to imagine something so horrible, so shocking, you may need counselling tomorrow. Imagine a world with no potatoes, no chips. Hand up if you're a bit distressed. Yes, see, I was told the Brits love their fish and chips, but don't worry, as far as I know, your potatoes are safe. But I'm sure you will sympathise with us Aussies when I say our potatoes came under threat. And you know, I know you can sympathise with us, just like when we very occasionally lose the Ashes. Our potatoes came under threat from an introduced pest from South America, the evil potato moth caterpillar. This caterpillar did damage to crops, but never fear, science came to the rescue. Scientists knew that in South America, the caterpillar had a natural enemy of its own, a parasitic wasp. This tiny black wasp injected her eggs inside the caterpillar. The baby wasps are like wriggling white maggots. They hatch out of the eggs and then eat the caterpillar from the inside out whilst it's still alive. Eventually, they burst out of the caterpillar like a scene from the movie Alien, form cocoons on its dying body, and change into adult wasps, continuing to save our potatoes. The cool thing is, these wasps are really specific, so each species of wasp can only attack one or two kinds of caterpillars. They can be used in crops without affecting the rest of the environment. Now, with 7 billion people in this world, being able to grow food crops like potatoes in the most sustainable and productive way possible is vital. So I'm a taxonomist. That's the scientist that names and describes new species. I'm working on a group of these wasps called the microgastrons, trying to work out how many species there are and what caterpillars they parasitize. But of course, when I catch a caterpillar in the bush, I can't just ask it what wasp is going to attack it. And when I catch a wasp, I can't ask it what caterpillar it attacks. So how do we work it out? One way is trying to use the DNA inside the wasp's guts. It's just like if you had a banana for breakfast, and then immediately afterwards you let me rip open your stomach and look at the DNA inside, I wouldn't just find your DNA, I'd also find the banana DNA. The same is true for the wasps. Eat the caterpillar, ingest the caterpillar DNA, and parts of this DNA can be used kind of like a fingerprint. Just like your fingerprints are unique to you and different for everyone else in this room, parts of the DNA are unique for each species of caterpillar. If we can match up wasp to caterpillar, we can start building our wasp weapons arsenal. In case we need to fight the caterpillars for our food in the future, we'll be able to win. Oh, and next time you eat a potato, have a thought for us Aussies and the wasps that saved our chips. Thank you.
So I think it's a really exciting idea. I guess I was wondering, historically, we haven't been very good at releasing pests and parasites to control things that we want to control. Do you think that's something we're getting better at?
Definitely, I think so. I think there's, especially in Australia, we've had some really horrible things. You might have heard of the cane toad incident. These days there's a lot more stringent controls. And the cool thing about these particular wasps is they're so specific. So they can only attack particular kinds of caterpillars, and it's to do with the fact they have a symbiotic virus in them, which actually suppresses the host, the caterpillar immune system, and it can only work on particular kinds of caterpillars. So they should be able to sort of not affect the rest of the ecosystem.
I love wasps. I'm actually doing an event tomorrow called What's the Point of Wasps? And
I saw it in the
which
program.
Which is brilliant. And one of the things I've been trying to pin down is the number of species of wasps. And it's very hard to get any sort of number. So I'm asking this from a purely selfish viewpoint. What number should I tell the audience tomorrow?
Oh, gosh.
What's the latest?
I can only tell you about my one subfamily, which is, it's thousands. It's, 2,000 or 3,000, and that's in one tiny subfamily. So,
yeah, hundreds of thousands.
Hundreds of thousands.
Yeah,
and,
okay.
We only know maybe like 15%. That's how many has been named.
So you can't get it wrong.
I can't get it
still.
wrong.
Yeah.
It's great. I'll just say hundreds of thousands, and I'm right on the money. That's perfect. Thank you.
So I have a follow-up question, if there's time. Do we have any idea why they're so specific? Like, what's the evolutionary benefit to just attacking one kind of caterpillar?
I guess parts of the reason would be so they're not competing with each other for the same hosts. So these particular ones, because of the virus in it, it sort of allows the eggs to survive in that host. As the butterflies and moths evolved and formed different species, we were able to have the wasps evolve and form different species as well. So different niches, we call it.
Okay, we spoke earlier about charisma. When the chips are down, she's got it in spuds. Erinn Fagan-Jeffries. And also at over 10,000 miles from Adelaide, she has come further than any of our other 26 Famelabbers. Now, Australia is not only the most distant participant in this year's Famelab, it is also the first alphabetically, and as luck/random number generator would have it, next we come to the country that's second, Azerbaijan, and their winner, Mirlan Karimov. Mirlan is a 19-year-old petroleum engineering student at Baku Higher Oil School. But if he wins through to the final tomorrow, he'll be a 20-year-old petroleum student because it's his birthday tomorrow. What he gets really excited about is physics, especially quantum physics. Milan says most of his spare time goes into learning more about it and attempting to uncover reasons behind the universe and why we're here. Can't promise he'll squeeze the ultimate answers to life, the universe, and everything into the next 3 minutes, but I'm sure he'll give it a good well. Please welcome our 2016 Famelab Azerbaijan winner, Milan Karimov.
It's so romantic, yes? After marrying, I'll take you to the stars. Yeah, presumably all, at least once in their lives, have gazed into the sky and pondered why the sky is so dark at nights. After Isaac Newton discovered the universal gravitational force, he put forward an argument that the universe should be infinite and fixed. Otherwise, it will collapse. However, according to Olber, if the universe had been infinite and unchanging, we would have seen the same number of stars in every direction in the sky at night, which means every single direction we glance in, we will see a star. And based on Olber's conception that the intensity and brightness of light coming from the star doesn't depend on the distance it covers, and admitting Newton's view that the universe is infinite, we may say that the intensity of light coming from an infinite number of stars will be infinite. In this case, apart from light night, we will vaporize as a result of infinite energy. However, it is not so. The sky is still dark and we are luckily alive. The reason for the dark sky is that the universe is expanding and changing. Stars are always moving farther from us. Hence, the expansion rate increases every time. And then, we know that when the body moves away from us, the light coming from that becomes redshifted. And this is called Doppler effect. Doppler effect works for sound waves as well. Think of an ambulance car which is driving away from us. The further it moves off, the lower the sound frequency becomes. Kinda du, 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 du. Now back to stars. The light, which is gradually getting redder, eventually becomes infrared, which is invisible to us. That is why we see just darkness instead of some stars in the sky at night. This is the first reason why the sky is so dark at night. And almost everybody here will say that nothing can move faster than the speed of light. But I say no. There is an exception. There is no restriction for the expansion rate of the universe. It can move faster than the speed of light. Therefore, the light coming from the distant stars where the universe moves faster than the speed of light doesn't reach us. That is another why we see just darkness instead of these stars in the sky at night. And to sum up, what you see when you look in the sky at night is compelling evidence that the universe is expanding. Thank you.
When did you meet the universe for the first time? When did you meet the universe? When you was child? When you get in love with physics and universe and stuff like that?
Yes, I met physics 2 years ago, and the last 2 years I spent my time on physics. And it's very interesting for me because I'm absolutely into finding the reason behind the universe, why we are here, why we came to, why we are sent to the universe. What is the reason for that? And I'm searching for that.
Okay, so simple. That's okay.
We started off with Quentin talking about quantum physics, which is really the physics of the very small. You've ended with talking about the physics of the truly enormous. Do you think we'll ever be able to reconcile those two things together?
Could you please repeat?
We started off hearing about quantum physics. Quentin mentioned your interest in quantum physics, which is the physics of the very small. You finished your presentation with the physics of the very massive, the universe. Do you think we'll ever be able to bring those two things together?
Yeah, I want to back to the quantum physics. For example, probably one would say that once upon a time, the universe was expanding more slowly than the speed of light. And in that case, there were no stars from which the light would reach us. In that case, the universe was light at night. Yes, it was. You're absolutely right. Here the question comes, where has that light gone to? This light is around us still today, but its wavelength has stretched so much that is why we are not able to see it. And it puts that the sky is not so dark in reality, but it's us mortals are not able to see this brightness. Yes.
Okay, expanding universe, expanding our minds, our Azerbaijani champion, Milan Karimov. Now, Milan's come over 2,500 miles to take part in Famelab International, and just under 500 more on the other side of the Caspian Sea is our winner from Kazakhstan, Dana Akilbekova. Dana is a researcher working on biophysics and tissue engineering, but as a kid, she says she was really shy, hated talking to strangers, and avoided anything where she was the center of attention, like, say, Famelab. Dana even says it was one reason she chose to become a scientist. It helped me a lot in hiding myself. All went well with this covert career until it came for Dana to apply for internships as part of her master's. Ignoring all calls and emails from potential employers and clamming up during interviews turned out to not be a very successful strategy. So Dana decided she needed to make changes, and she made big ones. She started telling people about her work. Then she joined a Toastmasters club in Kazakhstan. Then she entered Famelab. Then she won, and now she's here. Please welcome our stunner from Astana, Dana Akilbekova.
Ladies and gentlemen, let me introduce you a game changer. It can change lives of million people struggling with diabetes type 1. People with type 1 diabetes cannot produce their own natural insulin. They rely on daily injection of insulin to stay well. Without these injections, their blood sugar level gets so dangerously high it could be fatal for them. We scientists have been searching for ways to make blood sugar control easier and more convenient for patients. So that's where this guy comes in. So what it is, it's a micro-sized, well, imagine, it's a micro-sized delivery vehicle made of the polymer from the outside with the insulin delivery cells inside of it. Once it's injected into your body, it would start circulating your body and producing insulin whenever you need it. However, in creating this kind of vehicle, we scientists and engineers face a lot of different challenges, and one of them being biocompatible, meaning it won't be recognized and destroyed by your own immune system. So everything starts with injection of this vehicle. Your body not always knows what is good or bad for him. It triggers complex immune response involving interaction of various cells like mast cells, macrophages, fibroblasts, you name it. All of them have a huge arsenal to defend your body by attacking and destroying intruders. It lasts for weeks until they completely destroy it. So how can we help this little guy to overcome its challenges and trick your body? By changing the way your body interacts with it. It can be done by reprogramming or instructing your cells to change their behavior toward the vehicle, make cells more friendly. And that's what my research work is about. We engineer smart polymers and materials to affect cells directly and make them behave differently. We're able to see the changes how cells attach to the vehicles, ways they move around, and produce different proteins. We're able to see the, how cells attach to, by chemically modifying the surface of this vehicle, cellular response to it, give it optimized and tailor it. We're able to change cells' attitude, make them our friends. And now this little guy can do its work safely and properly. Thank you.
So do you think this is just useful for insulin, or are there other conditions that you might be able to treat with these kinds of polymers?
Excuse me, could you?
Do you think that the polymers that you're talking about have potential to treat other illnesses as well as diabetes?
Well, this is not about the polymers. I guess it's about the vehicle. So polymer is just a material. Why we use the polymers? Because they're not very harmful for your body. But yeah, it can potentially be used for delivering any kind of drugs, not just insulin, but for example, chemotherapy, any drugs for the tumor or for the cancer. Yeah.
How big in sort of actual terms is what you're
Well,
doing?
yeah, this is not microsize. It's actually a favorite toy of my one-year daughter, I guess. I hope she's not crying right now. Oh yeah, it's, well, in our lab, we are trying to make it around 100 to 200 micrometers. So it's about the size so it can enter actually the blood vessel, but it's big enough so the cells won't just engulf them and just destroy them right away. So that's our goal.
So it could be cool in the next year, have these kind of buses inside our body to help cure some illness. Can be a reality.
That's a very good question. Well, my professor thought that it is. I'm very optimistic as her, so my guess it's not a matter of 5 years or 10 years. It's probably 20 or 30 years when it's actually can come true. Yeah, we still have a lot of work ahead of us, and it's very exciting. So we can help the people.
I feel like it needs to communicate with the nanorobots. I think there could be some synergies.
Yeah, we don't believe in that.
Okay, I have to stop you there after some smart material about smart materials. A Kazakh standing ovation for Darnell Akelbekova. And finally, for now, for we are 8 down, 1 to go, although of course there's another batch of prime Famelab coming up right here at 4 p.m. in the third Tremini final. Finally, for now, we're going on a Qatari safari with our very first ever Famelab winner from Qatar, 3,300 miles from Cheltenham, and that is Ida Rafat. Ida is a chemical/environmental engineer working on wastewater treatment for ConocoPhillips, but says she's passionate about learning anything to do with science, from space exploration to evolutionary theory to quantum physics. Ida says she's blessed to live at a time where 90% of the accumulated knowledge of humankind is available at our fingertips. But which tiny fraction of a percent of that knowledge is she going to be able to deliver for us in the next 3 minutes? I don't know, but I hope she does. Get ready for some Doha hoo-ha with our very first Famelab winner from Qatar, Ida Rafat.
Innovation is a fundamental reason of our existence, survival, and evolution. In order for us to innovate, we had to utilize the materials around us. Our ancestors, for example, have moved all the way from the Stone Age to the Bronze Age to the Iron Age. And today, we ourselves are witnessing the Silicon Age. Because without the silicon, we wouldn't have all this mad revolution in electronics and communication. Now, looking at the future, what will be the next game-changing material that would revolutionize our society? Scientists may already have the answer, and it's a material called graphene. A fascinating fact about graphene is that everyone in this room have formed the graphene ever since you were in kindergarten and draw with your pencil for the very first time. This is because the material that comes out of your pencil is called graphite. Graphite is made of hundreds of thousands of flat carbon sheets that are stacked on the top of each other. Each sheet of these is graphene. So basically, graphene is the building block of graphite, our own pencil material. Graphene is found to be the best in everything. It is the lightest, thinnest, strongest material known to man. It's bendable, stretchable, transparent, and it's a phenomenal conductor of heat and electricity. All those extraordinary properties in a single material are the result of 3 unique attributes in the structure of a graphene. Number 1, this flat carbon sheet of a graphene is only 1 atom thick, which is basically the smallest building block of any material. And that's why graphene is actually called the world's first 2-dimensional material, because this sheet has a length and width and virtually no thickness. Number 2, every carbon atom in the graphene sheet bonds with 3 of its sister carbon atoms, forming a hexagonal network of incredibly strong and short covalent bonds. These bonds make the graphene the world's strongest material. So this 1 atom thick invisible graphene sheet is so strong that it can support the weight of a 4-kilogram cat. And finally, we're going to have a cat hanging on thin air. Number 3, Each carbon atom still leaves one extra unbonded electron that moves throughout the sheet with a speed approaching the speed of light, making graphene even more electrically conductive than copper. So one can only imagine the limitless future applications we can have from such a phenomenal material. From making it transparent foldable touchscreens, to stronger and lighter aircrafts, to supercapacitors, to even desalinating seawater. So the next time you draw with your pencil, remember that you're making your own personal fragments of graphene, that super wonder material that will probably draw back our own future. Thank you.
Graphene seems so a cool material, but what is the problem right now in using graphene?
What's the problem?
Yeah.
I would say that, yes, it has been a great breakthrough to isolate the graphene for the very first time. We have characterized it, we know its properties, we have tons of scientific publications about it, and now the obstacle is how can you produce it on a larger scale? And all scientists right now are trying to perfect the production and the synthesis process of how can we do it in a scalable manner. Because the two production process currently for to synthesize graphene is basically the mechanical exfoliation, which is the Scottish tape method, which is apparently not very scalable method, and the other which is the chemical vapor deposition, which is still people are working on how to perfect it so we can do it in a larger scale.
Yeah.
So what application are you most excited about? I like the levitating cats idea myself, but
well, I might be a bias because I'm in a water treatment, so I would say it's in desalination. To have like this one atom thick membrane system, it will be a really revolutionary thing, especially with the very recent studies that shows that it really can have a very high fluxes of water passing through it, and with very extremely 300 times more salt rejection compared to the conventional polymeric membranes that we're using, for example, in reverse osmosis. So yeah.
A big game changer in the modern world has been oil-derived plastics, and that's been a phenomenal thing, but also we see all sorts of pollution problems. Is there a downside to graphene? Is there any potential pollution
Yes,
problem?
actually, that's a good question. Like, people are all excited about all these properties of it, but we're still not really quite sure of what could be the environmental impact of it. And this is still a growing area of research. People are starting to understand what could be really the environmental impact of such 2D materials. They're still pretty new. They're only it has been isolated for 10 years right now, which is pretty new compared to the material life.
Okay, a fully three-dimensional explanation of a two-dimensional material, our mean graphene machine, Ida Rafat. And that is that. Well, not quite. They were all great, Famelab this evening. They've come an average of 4,452 miles each. Do I over-research this to take part? So it would seem cruel and mean and stupid to in any way split them up, to select 3 to go through to the final and 6 to not. But we've got the perfect people to do it. So judges, you need to be incisive, insightful, and inside 10 minutes. Can you give them a burst of applause to send them on their way, please, judges? Feel free to yell any names in the last chance to influence them. No, it's fine. Yeah. Right, fine. Now, while they're away deciding which 1/3 get through to tomorrow's final and which 2/3 get herded towards the Famelab exit, this is where you also collectively get to choose one of the 9 to go through. Now, as those of you who are here in the first part will know, we're trying a revolutionary new system of voting this time. And as you will also know, it failed. Now, there is an old saying in Britain, if at first you don't succeed, try, try, try again. I don't believe in that. If your system turns out to be pants, give up on it. So we're going to vote in a different way. We're going to vote by audience applause. And just to make it more interesting for you, and our Famelabbers don't know this, can we get all 9 of them, although I can see we have one absentee, if we can get 8 of the 9 at least, back up on stage, please, right now. What we've set up is a little clapometer. We're not going to rely on just the opinion of Helen from last time. We've actually got a clapometer set up at the back that will register your applause. So once we get everybody in position, I'm just going to count 1, 2, 3. 1, 2, 3. You can do it in order if you like. That will probably help. Okay, we're going this way round, are we? 1, 2, 3. Yeah, try and get your order right. It's going to help things. We'll spend 4 or 5 minutes swapping order around. This will be good. Fine, I'll just go in here. And that won't really help things at all as well. So I'm going to go through all their names one more time. And you can get some, just to remind you who they all are. So going from your right audience, my left. Hold your applause for a second. So we have Thatch Pham from Vietnam. Constantinos Timinis from Cyprus. Jackson Itakawa from Brazil. Eliska Selinger from the Czech Republic. Khao Thonsom Boom representing Thailand. Ida Rafat, hang on, we've gone slightly, have I gone, hang on, no, I think. Oh, well, maybe I just changed the order to fit it. I'll go with my order anyway. So we'll go back. So Erinn Fagan-Jeffries from Australia, Milan Karimov from Azerbaijan, Dana Akilbekova from Kazakhstan, and Ida Rafat from Qatar. Is there anybody whose name I didn't say in that? Good, that's fine. So the next time I say these names, we want your applause. Now, there's a tendency to clap slightly less at the start and build up, so resist that urge if you possibly can. Give it your full, and it's whoever you want to vote for. Are we all clear? Right, then let's go for it. I'm going to change the order randomly here. We'll start with Ida Rafat from Qatar. Right, it's the volume, not the duration. It's the clapometer. That's a top tip for you. Khao Thonsom Boom representing Thailand. Eliska Selinger from the Czech Republic. Jackson Itakawa from Brazil. Thatch Pham from Vietnam. Constantinos Timinis, our Cyprus winner. I've now lost my own place. Erinn Fagan-Jeffries from Australia. Milan Karimov from Azerbaijan. And if I've got this right, finally, Dana Akilbekova from Kazakhstan. And just as a control, Cunaguero from Manchester City. Yeah, fine, that's good. It's just a normal thing to calibrate the equipment. Okay, now, the other new thing we're doing this year, which is why I've also got everybody up on stage as well, is we thought, and you'll know this if you were here in the first half, in the first semi-final, is we thought we've got all these people from all across the world covering all these thousands of miles. They don't just know about their subjects. They might know some other stuff as well. So just while the judges are deliberating, this is your chance to ask them questions about science. Now, they're not a brains trust. They don't claim to know everything. You might ask a question and nobody knows. But you can either ask them about what they've spoken about or just generally anything to do with science whatsoever at all. So who would like to go first in this crucial part of the evening or afternoon? That looks suspiciously like a hand down there, but I don't think it was. There's a phone going off, but that's not quite the same thing. Oh, there's a hand there, right, and there's a hand there. We'll go to that one first.
My question is for Erinn and the study of...
My question is, how do you turn off a mobile phone quickly?
And potatoes, wasps, and caterpillars. Is the science being used on other agricultural products, and is it limited to Australia, or are there global studies being done on this?
Yeah, for sure. So I can only really tell you about the one group of wasps I study. They're called the Microgastrinae. They're a subfamily of wasps, of parasitic wasps, and they're used in a bunch of different crops. So the potato example is just one that's used in Australia at the moment. It's working. It helps the crops, the pests keep down. But there's heaps of others. So there was a wasp actually from the UK that was taken to New Zealand and helps control pests in crops of brassica, so broccoli and cauliflower, stuff like that. But there's heaps of examples worldwide. So yeah, just wherever there's a caterpillar pest, there's probably some sort of wasp that attacks it.
Okay, thank you. Remember, you can ask about anything at all. What is earwax? Is the ring of invariance of an algebraic group acting on a polynomial ring always finitely generated? Any old question will do. We'll go to gentleman there.
We had one talk on the DNA nanobots as a delivery mechanism for medicine and one on smart polymers. One of the points that the judges also picked up on was, is there any possible cross-fertilization between those two research tracks?
Suggesting interbreeding between Famelabers.
I didn't understand at all the question. Could you?
Yours was on the nanobots.
Yeah.
And there was also a talk upon using, that's right.
Yeah, Dana, yeah.
The question is, you're both talking about delivery systems for medicine. You're both looking at potentially delivering cures to specific cells and targeting medication. Is there any cross-fertilization between those two research tracks? Can you help each other?
So should you and Dana be talking to each other?
Yes. I think that it will be possible, but we need more research on that. So we can do that, and there are huge probabilities for nanobots, so they can interact with technology, with other technologies.
Yeah, thank you. Well, I guess we are a little bit like competitors at this point of, like, we are all scientists are competitors. Who's going to publish faster or do something faster? Well, personally, me, I don't really think nano something is actually very reliable method because it's quite expensive in your body. You don't want to put anything that you don't really see under microscope normal. At least you can see No, I'm kidding. He's just trying to kill me right now.
It's okay,
with his eyes.
everyone's already voted. It's fine to trash talk the other finalists.
No, I'm kidding. No, for my application, it's quite hard to do nano because we want to actually put insulin-producing cells inside. So it should be quite big. So it will be like insulin-producing islets. So they should be around 300 to 400 micron size. So you need to have quite a big polymer to put inside. But for drug delivery, I guess for cancer research, I guess nanos can work fine. Good luck with that.
Okay, so I think the answer is, in theory, these two disciplines could collaborate, but not maybe these two individuals, I think, in the near future. Right, who's next? If there is a next one, you can ask about anything to do with science at all. Any silly question you've always wondered about. Yes, gentleman there, thank you. I'm glad we ought to be, I imagine you're a curious audience with questions to ask. Don't be afraid of asking something daft, that's what we're here for.
So this question is for anybody really among the 9 of you. How big is the element of chance in finding a new scientific discovery?
So how much of science is down to chance rather than to deliberately narrowing fields round? Would you like to, do you want? Okay, so the question was, in any area of scientific discovery, when you discover something, how much of it is down to deliberately and painstakingly, I'm interpreting a bit here, deliberately and painstakingly working to find something, and sometimes how much of it is down to luck, to chance. So it's the Alexander Fleming discovery of penicillin or whatever, which are classic examples of luck. Yeah.
Well, I think most of the scientific discoveries are actually by luck. People in the science, like wherever they are in the labs, they don't really know what they're about to discover in the next few minutes. It just happens. And sometimes it's a collection of sequence of coincidence that happens that make them like really be at the edge of science at that particular moment, and they discover something. So I don't think like when you're about to discover something, you really know that you are about to discover something. It just happens by luck. That's my opinion.
It's like the luck comes from the creation of the universe, I think. For example, I can say example, Isaac Newton. It's said that he was sitting under the apple tree and apple fell down his head, and he discovered the universal gravitational force, but it is not. In that time, Isaac Newton was dealing with so many problems. He was already familiar with so many concepts of this gravitation and this universal gravitational force. But this apple just made him to complete this all in just one theory. And luck is something 10% of these discoveries, I think.
Yeah, I mean, you always get these stories. I mean, the Isaac Newton one may or may not be a myth. I've heard of these stories that it might be a myth. It might have been a pear tree. But there's also one that the guy who invented the cloud chamber was working really hard on it, and then he went to the pub and had some beer, and a peanut dropped into his beer, and he saw the trail of little bubbles going through the beer, and he thought, so there's a top tip. If luck doesn't work, beer will help.
Yeah, for example, another example, for example, Mendeleev discovered this table of elements. But it is not luck, because he was thinking of these elements, and when he was sleeping, the brain was working yet. And it came to his mind, this table, and it was not luck.
Okay, anybody else want to chip in on that? Was an excellent question. I thought we've got time for maybe one more. Who wants a nice climactic question? There's a hand pointing somewhere. There we go.
Hi, it's not a general question. I wanted to ask something to Erinn, but
that's okay.
For you, I really enjoyed your talk on wasps, and I was wondering, aren't wasps dangerous for humans as well? And I was wondering, if you attack the problem of caterpillars with wasps, you're creating potential problems for humans as well, I felt. And I was wondering if you think that it's the best way to tackle the caterpillars. You know, it's sort of like cleaning spilled coffee with toxic cleaner or something.
Yeah,
so
I guess this comes from what people's idea of a wasp is. So when you say a wasp, at least in Australia, I'm not sure here in England, people think of big orange and black stripy things that are going to sting you and hurt you and, you know, possibly even be fatal. But most wasps are not like that at all. They're normally much smaller, and most of them are parasitic on insects or other animals with an exoskeleton. So these particular wasps wouldn't be able to sting a human. They're really, really tiny, like a couple of millimeters long. And most of the wasps in the world are like this. We only see the big ones, because that's what we're used to seeing as humans, is the bigger things.
I'll give you a bizarre piece of parasitic wasp presenter trivia here. I was told that because we keep finding so many species of parasitic wasp, if you want to get a creature named after you, the best chance is, it's like most of the US presidents, film stars, actors, they've just got a bit desperate. What can we call this? We'll call this wasp Brad Pitt. We'll call this wasp, it's the best chance of having your name on a creature is a parasitic wasp. There you are, it's a fine bit of trivia. Okay, that is all the instant questions we've got time for. Can we thank our panel for being a panel temporarily? It's nice to have them united before they're about to be divided. Can we please bring back on our judges as well, please? Come on in. Well done. I hope well done. Hard done. So we will start, as is the custom, with the judges' 3 choices, all equal, but obviously you can't say 3 names at once, so when they get to that part, ignore the order. It has no significance. They're all equal. Mattia, before you disperse joy to sorrow in a 1-to-2 ratio, a little bit about how tricky it was making your decision, or what was involved, and maybe anything about what you thought of the 9 as a whole.
Yeah, first of all, it was an honor to stay with you guys, and we enjoy a lot your stories. It was not so tricky. I mean, they are good speaker, but we get to a solution in 8 minutes. So.
All right, not so tricky. So I always think it's nice to tell everybody they were all brilliant and it was really hard, even if it was really easy. But it's just it's Italian diplomacy. I bow to you.
Yeah.
Fine, okay. Okay, and did you find it kind of difficult knowing that actually you've got to send some people kind of home empty-handed? Because there's nobody here. There might be people here who deserve to win, but there's nobody here who deserves to lose.
No, absolutely. And I think one thing that we really thought as judges was that we really enjoyed all of the presentations we heard. So there was no point at which we were thinking, oh goodness, when's this going to be over? I learned a huge amount today on all sorts of random topics, and I congratulate you all on that. I guess for us, there was a few people who kind of rose to the top by, I guess, particularly the strength of their narrative through the very brief talks that you gave. That was one of the things I think that really played into our discussions a little bit, and also the strength of a good analogy and how well that can be used. Those were the
Okay, anything to add before we do the big reveal?
No, I mean, it was a very entertaining thing to watch. This is an incredibly difficult... Yeah, it's worked. This could catch on. It's an incredibly difficult format, standing up, basically naked on the stage with really nothing to support you, and you've only got a very limited amount of time. And as Liz says, one of the things that really struck us was the strong narrative of some of the presentations that you did right from the start. It was driving forward. We know where you were going with it, and that was superb.
Right, I think the naked on the stage bit may have been suggested by the T-shirt
Exactly.
ripping in the S&M presentation, but there was nobody actually naked. That's just in your imagination, Adam. That's fine.
That is against the rules, I believe.
I don't think we've actually written a rule about clothing in, but maybe you'll need to adapt it. Okay, I think in whichever way you want to do it, 3 equal names, time for the reveal.
Let me explain again. It's not a rank, just recap the best 3 for us. And first to say the names, let me just suggest to continue with your project. Don't give up, okay, guys? This is just a step, but life would be long, so a lot of challenge will be in front of you. So, the first name...
Don't let me stand out of your line. I'm standing blocking the audience from you. I'm going to stand behind you in a meaningful way, yeah?
Are you ready? So the first name was Constantinos Timinis from Cyprus.
Woo! Stay forward. You've earned your place at the front.
Ready for the second one? Aida Rafat, Qatar.
Woo!
I always dream to say last but not least. Erinn Fagan-Jeffries from Australia.
Woo! Okay, so those 3, congratulations, going through. But I'm about to be handed the golden envelope. I know nothing of the contents. This, I'm afraid, is where we see. Now, as I say, if your vote overlaps with their vote, that's the way it is. But if you've voted for somebody different, then we send through 4 people. And the winner is... It's that WASP woman again. Erinn Fagan-Jeffries is the audience vote winner as well. That is it. Can we, first of all, commiserate with our 6 unlucky losers, because everybody was brilliant. And like I say, they get the rest of the Cheltenham Science Festival off. They don't have to come up with a brand new presentation for tomorrow. They get to swan around, have a good time. In a way, they've kind of done the cleverer move there, I think, as well. Can we also thank our 3 judges, please? Mattia Crevellini, Adam Hart, Liz Tunbridge. Thank you for coming, especially to those of you who've been with us since 11 a.m. Only 2 more hours of this marathon to go. It kicks off at 4 p.m. It is worth being there just to see if I spontaneously combust before it's over. But it's even more worth being there to see the last 9 of our 27 Famelabulous Famelabbers. Once again, thanks to everybody on the stage, everybody behind the stage, yourselves for coming along. Can I ask you all to leave now, except the 9 Famelabbers for the 4 o'clock heat, because we want you on the stage. Thank you for coming, and congratulations to our 3 winners.